I need to do is I need to understand the structure of DNA. DNA's got quite a complicated structure, and in order for us to make a model of it, we need to have some understanding of it. We also need to understand that there's a lot of quite complicated chemical components to it, and therefore it's going to take a little bit, it's going to be simplified. So that's the main thing. DNA is a very complicated molecule, and we don't want to go into it in too much detail. Now, when we're building models, one of the important things that you need is a key, and I don't have a piece of paper with me at the moment, but I'll tell you what the components are in my key so that you can see how they come together to build a model. The most important thing that we've learned about chemical substances is they bond together, and then they usually bond in one of two main types of ways, with ionic bonds if they're charged, or covalent bonds if they are sharing electrons. And in all of the structures that are present in DNA, they share electrons. So they're going to be covalent bonds. Now, that means there's also another interesting type of bond, which is called a hydrogen bond, and it helps hold some of these things together. So I'm going to kind of use a few different types of things that are all a little bit similar, but hopefully will build together to give you an idea of how this works. So we're going to build a ladder, basically, and our ladder is going to be composed of individual units. Now, most of our polymers, our big long chain substances, are made of individual units that we just add together, and DNA is no different. So DNA has phosphates, these beautiful little berry bliss, nice and squeezy with a little bit of jelly inside. So what I'm going to do is I'm going to attach this. This is called a phosphate group. So a P for phosphate. Yes, P for phosphate. So pH. So there's my first part of my nucleotide. Now, attached to that is a sugar. So I need a sugar. So I'm going to get one of these nice little fruits, and I'm going to stick that together there. So now I have a phosphate and a sugar. Now, the third part of my DNA nucleotide is a base. Now, I've cheated a little bit here because I've got these little sour squirms. And the thing that's nice about these little sour squirms is they have two different colors. So this one has a green and this one has a purple. Now, I actually don't want that for my nucleotide, so I'm just going to break it. But you'll see why the squirms work so well a little bit later on. So what I'm going to do is I'm going to attach the sugar group like this. So the sugar, here's my sugar. It's attached to the phosphate. It's also attached to this. This I'm going to call a nitrogenous base. Now, a nitrogenous base is basically a particular chemical substance that's got nitrogen in it. With a lot of the organic components in our bodies, they're made of carbon, hydrogen, and oxygen. And a few of them also have nitrogen in them, especially proteins, which we'll look at a little bit later. So that's why I've got so this is now what's called a nucleotide unit. So I've got a phosphate, a sugar, and the sugar is attached to a base, and this is called a nucleotide. Lots and lots of these all linked together form DNA. Now, there's one more important thing about the base, this here. Do you know how many different types there are?
Is it 2?
It's not 2.
4?
It's 4, correct. It's 4. So there are 4 bases. Now, the 4 bases have long names: adenine, thymine, guanine, and cytosine. But we abbreviate that to A, T, G, and C. Now, I'm a bit of a George Clooney fan, so George Clooney is a nice, easy way for me to remember G goes with C, George Clooney. I haven't particularly got an A and a T favourite, but there's a lot of them around, and it actually doesn't matter how you remember it. Some people remember it with the G's and the C's are curved letters, and the A's and the T's have straight lines. But however you remember it, that's the only way they bond, and it's really, really important, and you'll see why later on, that we know how they bond and that we keep those bonds consistent when we're building our DNA. So that means that this first little base here must be either an A, a G, a C, or a T. Ava, give me one.
A and T.
Okay, so let's make this an A. All right. So if we make the green an A, then I'm going to make a second nucleotide. So that means I need my phosphate, I need my sugar, and if I've made an A, then I have to attach a T. So here's another nucleotide. So now I have two, okay? And these attach like this. So you can kind of just roughly see now, if I can get close enough, I'm holding the sugars, the bases are joined in the middle, and then there's the phosphate groups that are sticking up the top. That's how the nucleotides join to each other across to make the rungs of the ladder. Now, because they're not holding together too well, I'm going to get another little one of my squirms that I haven't broken, and I'm going to replace those two individual ones with one that's joined. So now I have a model that looks like this, okay? So the A and the T are joined together in the center. It's like the one that I would step on if it was a ladder. And then the sugars and the phosphates go up. The next thing I need to do is to put another rung on my ladder. Ava gave us the A and the T for the first one. This time I'm going to go with a G and a C. Yours is looking great there, Sophia.
Thank you, sir.
It's always more fun if you get to do this yourself. Now, because I already called this A and T, I'm going to call this one G and C. So you see the different colors. So they're different colors. So I'm going to put them along there like this. And now my DNA molecule, whoops, looks like this. Okay, I'm going to clear some space so you can see it. Ever had that feeling when there's too much on your plate? We've got phosphate, sugar, phosphate, sugar. We've got a base here, a T and an A, and a base here, a G and a C. And that's also attached to a sugar and a phosphate and a sugar and a phosphate. Now, the interesting thing about this is if I continue for a little bit longer, maybe if I see if I can make one more. I should now have 6 nucleotides, okay? Each of these is a pair, so that's 2, 4, 6. And if I'm able to lean it, if I carefully lift it, then what it will do is it'll start to twist. And the twisting is how we see the spirals nature of DNA. So DNA is a spiral staircase. It twists around, and you can see with these little bonds, I can get it to do some twisting. And that's one of the nice things about this model, is it allows us. So the thing with modeling is that modeling has limitations. Obviously, we're trying to deal with very, very tiny things, and very, very tiny things are very hard to show. And that's one of the reasons we model these things. They're all atoms, molecules, little tiny things linked together. But they do give us a little bit of an idea about what the structure of DNA looks like. Building a model that looks something similar to this was what won a couple of guys the Nobel Prize they completed the structure of this in 1953. And that was Watson and Crick, James Watson and Francis Crick. And they did a lot of that off some of the work that was contributed by Rosalind Franklin. They are some fantastic models. Guys, thanks for joining me in the kitchen to make some DNA models. Unfortunately, we're out of time. I'm hoping that I won't have to miss you guys tomorrow for the trials, but do please be patient with me. If you're having any problems with Education Perfect, let me know. Otherwise, enjoy your DNA models, and I'll hopefully catch you tomorrow.